Welcome back to Gut Secretions. In this next video, we will be looking at the role of bile in digestion. The acidic chyme from your stomach empties into your duodenum and it needs to be neutralised by alkaline secretions. And these alkaline secretions come from your pancreas and from your liver, so your liver secretes bile. If this acidic chyme was not neutralised, the lining of your duodenum will become damaged and you would have, for example, the formation of duodenal ulcers. Also, if your chyme was not neutralised and did not become alkaline, your enzymes would not be able to be activated within your small intestine and you would not be able to chemically digest your food. Bile is synthesised and secreted by the liver. It is secreted into bile ducts, shown here, that merge into a common bile duct. In between meals, there's a small duct called the cystic duct that takes the bile and stores it in your gallbladder, up here. And then when the bile is emptied into your small intestinal lumen, it passes down via your common bile duct, which merges with your pancreatic duct, and then empties the bile into your lumen via the sphincter of Oddi or the hepatopancreatic sphincter. So what is bile and what does it actually do? It's a yellow-green alkaline solution. The greeny-yellow colour is due to biliverdin, which is a green pigment present in bile. Biliverdin results from the breakdown of the heme component of your hemoglobin. We secrete between 0.7 and 2 litres of bile every day. Within this bile, shown in this table, we have bile salts that are derived from cholesterol. These bile salts help to emulsify fats in your small intestine. We have cholesterol because that's required to synthesize the bile salts. We have a phospholipid called lecithin. This combines with the bile salts to emulsify your fats. We have electrolytes and water, essentially in the same concentrations as your plasma, except there is a very high concentration of bicarbonate ions, which makes this solution alkaline and enables the fluid to neutralise the acidic chyme in the duodenal lumen. There are bile pigments within your bile, which are produced from the breakdown of heme in your hemoglobin and your myoglobin. We will now look at how bile emulsifies fats in your body. The average daily intake of fats or lipids is 70 to 100 grams per day, mainly in the form of triglycerides. These triglycerides are chemically digested via an enzyme called pancreatic lipase. Let's think about fats in your body. So you eat a fatty meal, you chew it up, you swallow it, it goes into your stomach, it gets churned up and mixed with your gastric secretions. Within the stomach, we end up with two layers. We have a watery layer of chyme and a fatty layer of large lipid droplets that sits on top of this layer of watery chyme. Now, I've mentioned pancreatic lipase. Pancreatic lipase is water soluble. This means that if these large fat droplets we have can't be broken down into smaller, tiny fat droplets, the process of fat digestion would be extremely slow. So it's really important that we are able to break down our large fat droplets into little lipid droplets. So the large fat droplets need to be emulsified or mixed with the watery chyme by using an emulsifying agent, which is bile in this case. So when you make your salad dressing, you put your vinegar and your olive oil into a container, and I have shown a glass bottle here. You shake your oil and vinegar, and this breaks down your olive oil into smaller fat droplets. If you actually stop shaking this salad dressing, the olive oil and the vinegar will again separate into their bilayers. So your salad dressing needs an emulsifying agent, which will allow these two liquids that are normally unmixable to mix together to form an emulsion. So within your gut, your emulsifying agents are your bile salts and the phospholipid lecithin. So your fatty chyme empties from your stomach into your duodenum of your small intestine. We have large fat globules on top of the watery chyme. First of all, we have contractions. Remember, we have those mixing or segmenting contractions within your small intestine. These will help to break down your large fat globules into smaller, what we call emulsion droplets. These emulsion droplets are between 0.5 and 1 millimetre in diameter. These emulsion droplets would coalesce, except we have our emulsifying agents. We have our bile salts and our phospholipids. You can see on the diagram on the right hand side, the bile salts and those phospholipids embed themselves around the outside of these emulsion droplets, preventing the emulsion droplets from coalescing. Also, the emulsion droplets have a net negative charge, which means they repel each other as well. We now have small emulsion droplets within the lumen of your small intestine. These emulsion droplets are surrounded by the bile salts and the lecithin. This actually causes a problem because we want to break down the triglycerides within the emulsion droplets, but the pancreatic lipase cannot actually get in to access the triglycerides because of the bile salts and the lecithin. So the pancreas secretes a cofactor that sorts this problem out. It's called colipase. So it's secreted from the pancreas at the same time as pancreatic lipase. The colipase is a bit of a bully. It comes along, pushes aside the bile salts and the lecithin, which then allows the pancreatic lipase to get into the emulsion droplets to access the triglycerides and chemically break them down into monoglycerides and fatty acids. The monoglycerides and fatty acids diffuse directly through the phospholipid bilayers of your cell membranes. In this case, it's the intestinal epithelial cells or the mucosal layer of the small intestine, and eventually end up in your bloodstream. Importantly, aiding in the process of absorption of fats, we get the formation of extremely tiny fat droplets called micelles. You can see they're 4 to 7 nanometers in diameter. So according to the concentration gradient of monoglycerides and fatty acids, we get the formation and breakdown of these extremely tiny fat droplets. These fat droplets contain monoglycerides, fatty acids, and they're surrounded by, again, shown in green, the bile salts and also lecithin. Inside these micelles and also the emulsion droplets, you would also have fat-soluble vitamins and cholesterol. We now need to work out what actually leads to the secretion of bile into the duodenal lumen. What triggers this process? On this diagram, on the left-hand side, we have a stomach containing brown chyme. This chyme is being emptied from the stomach into the duodenal lumen through, you can see, this will be relaxed. This is the pyloric sphincter. If you look at number 1 here, you can see the presence of the acidity leads to the secretion of a hormone called secretin, which is secreted from cells that are called enteroendocrine cells in the mucosal lining of the duodenum. The presence of fats in the fatty chyme emptying into the duodenum leads to the secretion of a hormone called cholecystokinin, which is abbreviated to CCK. Again, from enteroendocrine cells in the mucosal lining of the duodenum. These hormones, the blue circles represent secretin and the red circles represent our cholecystokinin, are emptied into the bloodstream. Up at the liver, what happens is that secretin triggers secretion of the bicarbonate-rich alkaline solution from the bile ductal cells. So that's number 3. The cholecystokinin, here we have our red dots of cholecystokinin over here, comes along, acts on the gallbladder and causes it to contract. And it also relaxes the sphincter of Oddi down here. So we've contracted the gallbladder, which is going to release the bile in association with ingestion of a fatty meal. And the cholecystokinin causes the sphincter of Oddi to be relaxed, which will allow that bile to be secreted into the duodenal lumen. You do get some contraction of your gallbladder via vagal or parasympathetic nerve activation, but cholecystokinin is the prime mediator of gallbladder contraction. I will now summarise the important roles of cholecystokinin in aiding with the emulsion and digestion of fats. Just remember, if you eat a fatty meal, you need cholecystokinin to aid with the digestion and emulsion of that fatty meal. So here I have a list of the prime functions of cholecystokinin in association with ingestion of a fatty meal and the secretion of cholecystokinin from the enteroendocrine cells in your duodenal mucosa. That cholecystokinin will contract your gallbladder, leading to release of bile. We want bile because it contains bile salts and lecithin, which will act as emulsifying agents and allow your fats to form emulsion droplets. Cholecystokinin relaxes or opens your hepatopancreatic sphincter or your sphincter of Oddi, which allows the bile to be emptied into the duodenal lumen. Cholecystokinin also acts on the pancreas, leading to the secretion of both pancreatic lipase and the cofactor colipase. Now, as fats digest slowly, cholecystokinin actually acts on the stomach to inhibit the churning of the chyme within the stomach, and the cholecystokinin will also act on the pyloric sphincter to constrict the pyloric sphincter. So inhibition of stomach churning and constriction of the pyloric sphincter will slow the emptying of the fatty chyme into the duodenum. So if you think of fat digestion, you need to be thinking of cholecystokinin. Okay, we've reached the end of bile secreted into your gut. You've learned that your triglycerides or your fats need to be emulsified. The emulsifying agents are your bile salts and your phospholipid lecithin. We have also learnt that your fats or triglycerides are chemically digested via pancreatic lipase and the cofactor colipase. And we looked at the roles of the gut hormones cholecystokinin and secretin in the emulsification and digestion of fats. In the next video, we will be looking at pancreatic secretions and their roles in digestion.